All right, so welcome to the third and final video for our um, genetics and inheritance chapter. With this video, we're going to talk about some examples when inheritance deviates from the typical dominant recessive, so complete dominance inheritance pattern. And one of the first ones we're going to talk about is incomplete dominance. And so incomplete dominance is basically when you have an intermediate form between the dominant and the recessive trait. So for example, let's say we're looking at hair. Let's say the dominant trait is straight hair, the recessive trait is curly hair. So the intermediate form, the heterozygous form, is going to be wavy hair. Another example is flower color. So let's say the dominant trait is red, the recessive trait is white, so the intermediate, the heterozygous trait, would be pink flowers. There's also a form of cholesterol association where if you have the dominant trait, then you have normal cholesterol. If you have the recessive trait, which is hypercholesterolemia, which is where you have very high blood cholesterol, then you have 6 times the normal cholesterol level, which means you actually can have heart attacks as a child. But if you're heterozygous, you have an intermediate form, so you don't have as high as having the recessive, the homozygous recessive form, but you have a lower amount because it's combined with the dominant. So you have about 2 to 3 times the normal cholesterol level instead of 6 or greater times the normal cholesterol level. And so here you see that a straight-haired individual has offspring with a curly-haired individual, then all of their offspring will have that intermediate trait, which will be wavy hair. Now another example that deviates from codominance, or I'm sorry, from complete dominance, is codominance. And this is what we actually see with our blood types. And so basically, if you have those traits, they're equally expressed. So A and B are codominant with one another. This is how you can have the AB blood type. So for example, if under normal scenarios, if A was dominant and B was recessive, if you have an AB allele, then that would be an A blood type. But because A and B are codominant with one another, when you have the AB genotype, you actually have the AB blood type. And so if you're heterozygous for those, then you express both antigens on the blood cell. And so this is the case for the sickle cell gene as well, which we'll talk about here in a second. But just looking at the blood type, so if you have type A, you only have A alleles, right? The O allele doesn't express any antigens, so you actually can be AO and express only A antigens, or you can be BO and express only B antigens. But whenever you have the heterozygous form, which is AB, then you're expressing both A and B antigens, so you are codominant. So you're expressing both A trait and B trait. With the sickle cell gene, the different alleles affect the shape of the blood. And so HbA encodes for normal hemoglobin, HbS encodes for sickle cell hemoglobin. So if you're HbA dominant, right, so if you're HbA homozygous dominant, then you have normal blood. If you're HbS homozygous, I shouldn't be dominant, but if you're HbS homozygous, then you have sickle cell blood. However, if you're heterozygous, so HbA and HbS, so you're heterozygous, then you actually have some of the sickle cell trait and some normal hemoglobin. So this individual makes both types of hemoglobin, but they don't see the symptoms from sickle cell like a person that's homozygous for sickle cell. If they are homozygous for sickle cell, then their blood cells actually crystallize if oxygen slightly decreased, causing the red blood cells to bend in those crescent shapes. And because all of their blood cells have that trait, then they get significant side effects from that and significant symptoms. And that one of those is that it can lead to organ damage because of the drop in oxygen concentration, because um, the sickle cell shaped blood cells don't bind oxygen as readily. Um, and so basically what you can do, you can be a carrier of sickle cell. You have the trait, but you don't have the symptoms. So you're heterozygous, and then you can be homozygous sickle cell or homozygous normal. And so here's just a video showing both of those incomplete dominance and codominance situations.
In this example of complete dominance, a mother and her child have different alleles for the genes involved in one type of albinism. The mother is heterozygous, while the child is homozygous recessive. Both the mother and father of this child are heterozygous, so their gametes have an equal chance of carrying a dominant or recessive allele. Their children have a 25% chance of inheriting both recessive alleles and having albinism. With complete dominance, individuals who inherit either one or two dominant alleles are normally pigmented. How does complete dominance work at the molecular level? In this example of albinism, the dominant allele codes for an enzyme called tyrosinase, which converts the amino acid tyrosine into the brown pigment melanin. With either one or two copies of the dominant allele, enough tyrosinase is produced to generate normal amounts of melanin. Homozygous recessive individuals lack a functioning tyrosinase enzyme and produce no pigmentation. Complete dominance is not the only possibility for a heterozygous genotype. Sickle cell hemoglobin provides an example of a pattern of inheritance called incomplete dominance. A typical red blood cell carries between 200 and 300 million molecules of hemoglobin, the molecule that binds and carries oxygen molecules. A red blood cell filled with normal hemoglobin, designated HbA-HbA, is a biconcave disc. In sickle cell anemia, designated HbS-HbS, most red blood cells appear normal when the oxygen content of the blood is high. However, as oxygen levels drop, such as when red blood cells deliver their oxygen to the body's tissues, the cells become sickle-shaped and tend to clump together, clogging capillaries and depriving tissues of oxygen. The shape results from the abnormal hemoglobin molecules sticking together in long polymers, which does not occur with normal hemoglobin. People with sickle cell anemia usually die at a young age. Heterozygous individuals are said to have the sickle cell trait. The normal allele shows incomplete dominance, meaning that the expression of the heterozygous condition lies somewhere between the two homozygous states. People with sickle cell trait are generally healthy, but sickling and clumping of red cells may occur if there's a prolonged drop in the oxygen content of the blood, as might happen during travel at high elevations. Because normal hemoglobin molecules reside in the same red blood cell as abnormal hemoglobin, polymerization does not occur very readily. If two individuals with the sickle cell trait reproduce, their children will have a 25% chance of having sickle cell anemia, a 50% chance of having sickle cell trait, and a 25% chance of having normal red blood cells. This pattern is typical of incomplete dominance. In another pattern of inheritance called codominance, the effects of both alleles are separately apparent in a heterozygote. For example, the AB blood type arises from the presence of a type A polysaccharide and a type B polysaccharide. Two alleles of the same gene, IA and IB, code for different enzymes, which produce the A and B polysaccharides respectively. Populations have the IA and IB alleles, as well as a third allele called little I, which doesn't code for an enzyme. People with a homozygous recessive little I genotype have type O blood, which lacks the A and B polysaccharides. Blood type A results from a genotype of IAIA or IA little I, and blood type B results from genotype IBIB or IB little I. Note that while the IA and IB alleles are codominant to each other, each is completely dominant to the recessive little I allele. Knowing the patterns of inheritance of blood type alleles, we can make predictions about the outcome of a genetic cross. If a man with type A blood of the genotype IA little I and a woman with type B blood of the genotype IB little I had children, those children would have an equal chance of having blood type AB, A, B, or O.
All right, and so that video did a pretty good explanation or a summary of codominance and incomplete dominance. The next thing we're going to look at is referred to as polygenetic inheritance. So basically when the phenotype, the trait, so what you see in the individual is actually determined by multiple genes interacting with one another. Some examples of some examples of polygenetic traits are things like eye color, height, body size, body shape, skin color, etc. And these polygenetic traits are usually in a distribution within a population. So kind of like a bell-shaped curve where if we were looking at height, you would have short, tall, and then kind of your, intermediate, I guess we could say, height ranges, with very few individuals being short, very few individuals being extremely tall, and then a majority of your population falling in those average height ranges. So for example, here we have variations in eye color. You can have variation between the colors, which are blue, green, brown, hazel, etc. But then you could even have variation within those colors. So like light blue, or light green, dark green, etc. And again, those are determined by the combination of genes. And so let's say that height is determined by 3 genes. And those genes can have 2 versions of alleles. So uppercase A and lowercase a. If they are all homozygous dominant, then they will be tall. If they are all homozygous recessive, then they will be the shortest out of the population. And then any combination between makes you either be on the short end, the taller end, or the absolute average if you're heterozygous for both of those. And so again, you have this distribution of that bell-shaped curve that's determining what your phenotype is going to be based on the interactions of all of those genes that kind of account for that phenotype. And so what I also want to mention is that the genotype is going to affect your phenotype, so the way you look. But it's not just determined by genotype alone. Environmental factors can influence the phenotype. So for example, what you eat affects your height and your body size. So your genes can determine if you are, skinny, heavier, big boned, etc. All of the different ways we describe our body size or body shape. But the environment is going to affect our body size as well. So basically what we put into our body, what we eat, is also going to determine our body size. If we have a healthy diet, then we're going to be, we'll have a lower body mass index. Basically, if you have a non-healthy diet, then you can fall on the obese end, etc., regardless of what your genes are telling your phenotype to look like. And we see that in a lot of cases where the environment can alter what your genotype or your phenotype actually looks. There are also genes that can be linked together, and so they can be inherited or not inherited together. And so linked alleles are basically when they're located on the same chromosome. So for example, we were talking about earlier, we were looking at hairline and attached ears. And we said that if you have the dominant allele, it doesn't mean that you're getting, so if you have the dominant allele for free earlobes, it doesn't mean you're getting the widow's peak. However, if they are on the same chromosome, so let's say you have your earlobe gene and your hairline gene on the same chromosome, then they do carry with one another. When they're not linked is when they're on different chromosomes. So for example, you have your E, and then you have, let's say, your W. These are bad representations of chromosomes. So this chromosome can go one way, and then this chromosome can go the other way. But when they're on the same chromosome, they travel together and they're linked, and so they can be inherited together. But if you remember, crossing over happens in meiosis, and crossing over is when you graft genes, so they actually can be separated. So let's say this portion's grafted onto another gene, say that has the lowercase e, and so now you have a lowercase and a capital, and so that can actually cause that crossing over can cause them to be switched and not linked anymore. There are also inheritance patterns that are associated with sex-linked inheritance. So basically your X and your Y chromosomes, so your sex chromosomes. And remember, your sex chromosomes is that 23rd pair of chromosomes, and they're not homologous because they are X and Y. Okay, so your males will have one X and one Y, while females will have two X chromosomes. So with males, 50% are carrying X gametes and 50% are carrying Y gametes, where females will always have an X gamete. And so the male is actually determining the gender of the offspring. It's not the female. So if a male gives a Y chromosome, then the offspring will be a boy or a male. If the male gives an X chromosome or an X allele, yeah, an X chromosome, sorry, then the offspring will be female, because the female is always giving an X because they only have X's to give. And so you can have sex-linked genes where they're located on the sex chromosome. So these genes are only found on the sex chromosomes, and some of the more common ones are X-linked chromosomes, so genes that are only on the X chromosome. And so this means that with these sex-linked genes, they're more likely to be on males than females, because the male only has one version of that X gene. So for example, your male has an XY. And so if the male has the allele on the X chromosome, then it's expressed, right? So let's say color blindness, or let's do hemophilia. So let's say hemophilia is recessive, right? So the male can only have these two genotypes. So they have the X that has the dominant or the X that has the recessive. If they have that recessive allele, then they have the disease. Whereas females can have three combinations. They can be homozygous dominant, they can be heterozygous, or they can be homozygous recessive. So the males are going to be either or. They either have it or they don't. A female cannot have it, she can have it, or she can be a carrier. So that means the females have a less chance of actually expressing the gene than males. And so what we see with that is, let's say you have a female that's a carrier for hemophilia, and then the male that's normal. So the female offspring are not going to have hemophilia because they will get a dominant X always from the male. So that means they will always have that dominant allele, because the male only gives off the dominant allele. Whereas the X from the female is going to be dominant or recessive. So the females that are in the offspring will either be homozygous dominant or heterozygous. So they'll either be normal or they'll be a carrier. The male offspring has a 50% chance not to have it, because they will get the Y from the male and the dominant from the female, so they're a normal male, or they get the Y from the male and the recessive allele from the female, meaning that they have that disease. Okay, and so again, that's why the males have a higher chance of getting that sex-linked trait versus the females. So here's another
Various human genetic
animation video showing sex-linked traits.
disorders are said to be X-linked, which means that the defective allele responsible for the condition is carried on the X chromosome. This produces a particular pattern of inheritance because females have 2 copies and males only 1 copy of the X chromosome. X-linked characteristics are therefore expressed more often in males than in females, and as a consequence, X-linked diseases are expressed in more male than female victims. Red-green color blindness is an example of an X-linked recessive disorder because color blindness is not expressed if a functional allele is present. On the Punnett square, we can denote the X chromosome with the normal, functional allele with a black X, and the X chromosome with the defective allele with a red X. The possible progeny are XX, both normal alleles, red X, black X, carriers with 1 normal allele, black X, Y, normal males, and red X, Y, colorblind males. Note that the genotype must be either red X, female, or red XY to express colorblindness. A female of genotype red X, black X has normal vision and is a carrier for the disorder. Although she will not express the recessive trait, she can pass the defective X allele to her offspring. As this example illustrates, the chromosomal makeup of males makes them more susceptible to X-linked recessive disorders than females. For a daughter to express colorblindness, she must inherit 2 recessive alleles, genotype red X, but a son who inherits a single recessive allele will have the colorblind genotype red XY. As a consequence, colorblindness is much more common in males than in females.
Okay, and so that's sex-linked traits. Now, we can have what's known as sex influence traits. And so these are affected by the sex genes. And so, for example, sex influence traits are genes encoding these traits are located on the actual autosomes, so not the sex chromosomes, but the expression of that trait is affected by the testosterone and estrogen, which comes from the expression of genes from the sex chromosomes. So while these genes are not on the sex chromosomes, their expression is tied to the presence of those sex chromosomes. And so, for example, baldness is several genes that influence hair patterns on the organism, but is also influenced by whether or not there's the presence of estrogen or testosterone or lack of presence. And so it's influenced by the sex chromosomes themselves, and so it's a sex influence trait. Now, some issues that can come up with chromosomes is that there can be alterations in chromosome number or structure. And so non-disjunction can happen during meiosis where you don't have the alleles to separate appropriately, or I'm sorry, the chromosomes to separate appropriately. So in meiosis, you could have the homologous chromosomes to not separate, or the sister chromatids to not separate. And depending on whether you have non-disjunction during meiosis 1 or meiosis 2 can determine how many of the gametes are being affected. And so a gamete, because of this, could end up with 2 copies of a chromosome or no copies of a chromosome. And so they'll have extra copies or they'll have no copies. And so some examples of non-disjunction lead to trisomy 21, which is Down syndrome. So they basically have an extra copy of the 21st chromosome, or you can get alterations in those sex chromosome numbers. So here you have regular meiosis where the homologs split and then the sister chromatids split to give you one copy of each chromosome. Now, if you get non-disjunction at meiosis 1 and those homologous chromosomes don't split, one cell will not have that chromosome and another cell will have an extra copy. And so when those sister chromatids split, you have extra copies in 2 of the 4 gametes and no copy of that chromosome in the other 2. But now if meiosis 1 happens appropriately and you have a non-disjunction at meiosis 2, 2 cells are normal. One cell has an extra copy, one cell has no copy. And so Down syndrome comes about because of the extra copy at chromosome 21. So you get 3 copies. So it's called trisomy 21. And Down syndrome has distinct physical features, developmental disabilities that we see that's very common with individuals that have this disease. And this actually happens pretty commonly in 1 in 1,000 live births. Risk of trisomy 21 increases as you get older. You have more of a chance for that non-disjunction happening, and you can actually do genetic tests while the embryo or while the fetus is growing to determine if your offspring has extra copies of the 21st chromosome. You also can have alterations in sex chromosomes, so your non-disjunctions happening with the 23rd pair of chromosomes, and you can get varying combinations. So for example, you could have an extra Y or an extra X, or you could have no Y and only one X. And so if you're XYY, it's known as Jacob's syndrome. The offspring are males. They tend to be tall and pretty normal. Klinefelter syndrome is a double X and a Y. So these are males, but they tend to be sterile. They have some mental impairment and breast enlargement because they have that extra chromosome. And so that X chromosome is being expressed doubly, whereas originally in a male, you only get expression of one X gene instead of 2. And then Turner syndrome is where you have no Y. So because you don't have a Y, you typically only have the female characteristics. And so they tend to be shorter and sterile because of the lack of the second X chromosome. So here's a chart just showing some genotypes for alterations of chromosome numbers. What the phenotype is in terms of sex, the common name for it, and then the frequency of births in which that actually happens. Now, some other alterations of chromosomes can be things like deletions, so where a piece of the chromosome is broken off, or you can have translocations, so where a piece breaks off and then attaches to another place on that chromosome or attaches to a different chromosome. And again, these can cause big problems in the proteins that are coded for those segments, because if you delete a segment or add a segment, it's going to change your codons, which changes your amino acids, and it changes the proteins that are made. And now a lot of disorders that exist are revolving around recessive alleles. So a lot of genetic disorders come from being homozygous recessive, so recessive alleles. So if you have that disease, then you have to get one recessive allele from each parent. And so those parents are carriers. They don't show symptoms, but they are able to give an allele. And then if the offspring has an allele from each parent, then they get that disease. So for example, PKU, where you don't have an enzyme that can break down phenylalanine. Because of that lack of breakdown, it could lead to mental retardation. And so the way you treat it is you limit the phenylalanine in the diet. Tay-Sachs disease is another where it lacks an enzyme to metabolize a specific brain lipid, and you could actually lead to fatality by age 4 because it leads to brain dysfunction. So those are just some examples of recessive allele diseases. Now there are diseases that can be caused by dominant alleles, and these are usually dominant lethal alleles. And so if you are an individual that has a dominant disease or it's in your family, you're most likely heterozygous, or that person is a heterozygous. And so they have this kind of sort of genotype. The homozygous version of a dominant um, disease is usually fatal before the organism's born, because you have the expression of the gene, you have twice the expression of the gene because you have 2 of the alleles, and so that expression is usually enough to kill the organism. But whenever you only have one version of that allele being expressed, so one copy of that dominant allele, it's generally um, um, not lethal um, until after the organism has been born, and so the embryo has been born. And usually it's not expressed until midlife, unless it's something like um, Huntington's disease, where it's no, yeah, sorry, I don't know what I'm thinking. Usually not expressed until midlife. There we go. And so it's always lethal, so later in life. And the reason it persists in the human population is because of this midlife, right? So basically, it kills the individual after they've reproduced. And so what that means is they have a chance to pass on that dominant allele. And so they have children before the symptoms occur, and so they don't even know that they have the disease to decide whether or not they want to have the chance to pass on that allele to their offspring. And so each child has a 50% chance of inheriting the lethal gene because it is a dominant disease. So if it was recessive, it would need both recessive alleles. But since it's a dominant disease, it only needs that one dominant allele to be passed on by the parent. So let's say if the mother did pass on the recessive allele, the father could pass on the dominant allele, and it really wouldn't matter what this is at that point. In terms of being affected by the disease. And so one thing I just kind of want to finish off with is that we've talked about genes, we've talked about the expressions of the genes, and the genes are coding for proteins, not behaviors, right? And so the genes tell specific proteins to be made, and the proteins have specific functions that lead to all of the characteristics that we talked about. And so those proteins are like making hormones or enzymes or neurotransmitters or messengers that travel across the body that lead to the specific phenotypes that we talked about. And so the expression of a particular hormone or enzyme causes the coloration of our skin to be a certain way, or our hair color, or our height, because you either have a lot of expression or low expression of that protein. And so that determines the phenotypes that we look at, right? And so one big thing to just know is that genes are coding for those proteins, not for specific behaviors. The proteins are kind of what determines that outcome, okay? And so that's it for the genetic and inheritance chapter, and that's it for all of the information for this course. So I really enjoyed teaching you all. I really kind of enjoy teaching this class. It's a lot of interesting topics, a lot of interesting subjects, and I hope you've been able to benefit from this. Hopefully you've learned something, and that you can find something useful from the class as you move forward in your lives or your careers. And I just wish all of you the best. If you ever have any questions or any concerns dealing with academics, or questions about college or something outside of the class, feel free to shoot me an email, even if it's after your term. I'm always glad to help students in their academic journeys. And I want all my students to do well, not only in my class, but just to be successful in life. And so with that, thank you for letting me teach you. Um, let me know if you have any questions, any concerns, and as always, you can reach me through email and discussion. And good luck on everything that you do, and hopefully we will talk to you later or see you around. So have a good day.